Okay, students, welcome to Chapter 9, which is going to focus on primarily skeletal muscle. We're going to go through kind of the anatomy of it and then generally the physiology. So that's where we're headed with this chapter.
Nearly half of your body mass consists of one primary type of tissue, muscle tissue. When you think of muscles, you likely think of the biceps of a weightlifter, but muscle tissue is also found in many other areas of the body, including the walls of the heart and stomach. There are three types of muscle tissue: skeletal, cardiac, and smooth muscle tissue. Skeletal muscle tissue forms skeletal muscles that attach to joints and bones and are responsible for overall body mobility. As a clinician, it's very important that you have a solid understanding of muscle tissue so that you can properly treat your patients with tissue and muscle injuries. For example, many patients pull or strain their muscles while playing sports. A strained muscle occurs when the muscle has been excessively stretched or possibly torn due to overuse of the muscle. The injured muscle becomes painfully inflamed, and surrounding joints are usually immobilized. As a healthcare professional, you will need to know how to treat these types of injuries. The standard treatment, known by the acronym RICE, which stands for rest, ice, compression, and elevation. This treatment helps relieve a patient's pain and decrease inflammation. A thorough understanding of skeletal muscle tissue will enable you to help patients recover from painful muscle injuries.
So as the chapter said there, the little video, it is focusing primarily on skeletal muscle, although we might talk a little bit about smooth muscle too. The third muscle type, cardiac muscle, you will discuss more extensively when you get to the cardiac chapter in A&P 2. The kind of one thing that muscle tissue, or really the two things that muscle tissues have in common, no matter what type of muscle tissue they are, is their ability to turn this chemical energy, to take the bonds of ATP and break them, and to turn them into some sort of mechanical energy, whether it's the movement of the whole body, the pumping of the blood by the heart, or say food through our GI tract if we're talking about smooth muscle. Muscle tissue is also very well vascularized, all three of them. They require a ton of oxygen and nutrients to do their job, especially skeletal and cardiac muscle. So there's a nice table in your book. It's table 9.3, and it really is a nice comparison between all these muscle types. So any kind of compare contrast types questions, that's a good resource for you to look at. We've seen some of these before, these prefixes, myo, mis, but one I think that's not on your initial list is sarco. All of these things are prefixes for muscle tissue, and so whenever we see any of these types of prefixes, you have to know that we're talking about muscle tissue. So the other thing that all muscle tissues share is these special characteristics. Again, it doesn't matter if it's skeletal, cardiac, or smooth muscle. All muscle tissues are excitable, right? They can receive and respond to stimuli. Skeletal muscle, for instance, gets a signal from the nervous system, and then you can contract your muscle and, straighten or your arm or move your arm or speak or anything like that. Contractility is the ability to shorten. Literally contract. That's what contract means. It means to get shorter. And so when muscle tissue is stimulated, it will get shorter. But it is also extensible. Muscle tissues can stretch without being damaged unless they're overstretched, as in the video was talking about, because they have elasticity. Not only can they stretch, but they can recoil back to their resting length as long as they're not overstretched, and then we end up with some damage. But as long as we're not overstretching our muscles, because of their extensibility and their contractility, they can go back to their resting length without being damaged. So these four characteristics are shared by all three types of muscle tissue. So most of this chapter is going to focus on skeletal muscle. So that's where we're going to start. It's talking about the blood supply and the nerve supply to skeletal muscle. As I mentioned earlier, there's a huge demand for nutrients and oxygen. Skeletal muscle, anytime you move, anytime you talk, anytime even your postural muscles that allow you to stand upright. And so not only do they need a ton of oxygen and nutrients, they also create a huge amount of waste by going through these contractile processes and physiology that we're going to talk about. And so every skeletal muscle gets an artery, a nerve, and several veins that'll drain it. And typically they'll kind of come in a bundle very near the kind of central part of the muscle cell, and then they branch through these connective tissue coverings, these sheaths that we're going to talk about in just a second. So this is what a whole muscle looks like, and this happens to be your quadriceps muscle, which is your big thigh muscle, because there's your femur right there. And so you can see that muscle fibers, or muscle cells, muscles in general, are really just these bundles upon bundles upon bundles of fibers. And in fact, one muscle fiber is the same thing as saying one muscle cell. And all of these kind of groupings of fibers get covered in these connective tissue membranes that really kind of help support the cells, help kind of reinforce the muscle itself. So if we start at the most kind of microscopic level, a single muscle fiber or a single muscle cell gets covered in a membrane that we call endomysium. It's areolar connective tissue, so it's very loose. Lots of stuff can move through it real easily. Around every single muscle fiber, you're going to find that. When you get several muscle fibers bundling together, it's a group then called a fascicle, and the fascicle gets covered in another membrane called the perimysium. This is now dense, irregular connective tissue, so still a fairly loose connective tissue. If you then bundle several fascicles together, then you get a whole giant muscle, and that then gets the epimysium membrane, which is more fibrous connective tissue, and sometimes will help connect the muscle to the bone. So you can really see it's this kind of fibers within fibers within fibers within fibers, all of which are surrounded with muscle membranes. In terms of attachment, remember that we have two primary attachments, at least two. The insertion, which is the attachment to the movable bone, and then the origin, which is the attachment to the immovable or less movable bone. And these types of attachments are going to be of two types. They're either going to be a direct attachment, where the epimysium, that outer membrane, actually gets fused to the periosteum of the bone or the perichondrium, the cartilage, so it's a real direct connection, or these indirect connections, which are kind of more common, which are these connective tissue, dense, regular connective tissue, and they come in two forms, the rope-like tendons or these kind of more sheet-like flat aponeuroses. But these are connective tissue covering, or connective tissue connections between muscle and bone. When we look at a single skeletal muscle fiber, remember skeletal muscle is striated. They are very, very, very long. A skeletal muscle fiber, for instance, in your thigh attaches up at the top of your femur to your hip bone and will go all the way down and attach again at your knee, and that fiber will go the whole length of your thigh. They are multinucleate, so you'll see lots and lots of nuclei. They have a sarcolemma, which is just a plasma membrane. They have cytoplasm inside of them. We call it the sarcoplasm instead because it's a muscle cell. And they have some granular glycosomes. Glycogen is our storage form for glucose, and so since our skeletal muscles use so much glucose, they can store it themselves just like the liver cells do, and then rather than hemoglobin being our oxygen carrier, muscle cells have huge conglomerations of myoglobin, muscle globin, and again, that's really just for oxygen storage. It's very similar to hemoglobin. You'll also see we have these striations, these stripes, the dark A and the light I. Now we're going to really see what actually causes the striations in skeletal muscle and cardiac muscle. And the reason we have that is because we have these little fibrils. So every single muscle fiber, here's a single muscle fiber. Here's our little sarcolemma plasma membrane, typical phospholipid bilayer plasma membrane. But inside these muscle cells, muscle fibers, are these myofibrils, and there are these rods, really, that make up most of the volume of the cell. And inside the rod, every little myofibril here contains our sarcomeres, and the sarcomeres are this very specific arrangement of our myofilaments, our muscle filaments, contractile filaments, and since they repeat in this sarcomere pattern, that's why we get these kind of dark A and light I striations that we see in skeletal muscle and cardiac muscle. So cardiac muscle has sarcomeres as well. So in terms of the myofilaments, we have really two main contractile proteins. We have the thick filaments, which is composed mostly of myosin. Myosin has basically like a tail form and then a head form, so you'll see that in just a second. And then we have the actin filaments, which are kind of more, they kind of twist together and make almost like a rope-like strand, and bound to that rope-like strand are two little regulatory proteins, trypamyosin and troponin. And you'll see kind of how this works and how this looks, and you're going to actually get a chance to really build yourself a model as well. When the myosin heads attach to the actin, that's called a cross bridge, and that's where our contraction of our muscle starts, is forming these cross bridges, these attachments between the myosin heads and the actin filaments. So here you go, here's what I was kind of talking about. So here's your big thick myosin head, your myosin filament, and you can see these little heads sticking off of it. So the tails make up kind of the filamentous part, and then the heads kind of stick up. You can see here's our thin filament, the actin units kind of twist around each other, and then twisted around the actin filaments are the trypamyosin, which is this one here, which blocks the sites on actin for myosin to bind, and then the troponin, which kind of locks it into place. When we look at a sarcomere, this is our contractile functional unit. This is what is responsible for the contraction of our muscles. Because of this very specific arrangement, we get these striations. So the A band is where the thin filaments and the thick filaments basically overlap. In the middle of the A is something we call the H zone, where we pretty much only have thick filaments. And then on the I, we only have the light filaments, the thin filaments, and they're kind of bound on either side by something that we call a Z-disc. And so we get this arrangement of this sarcomere that is really responsible for these striations that we see. So here's kind of what I was talking about. Here's a sarcomere. So one sarcomere extends from Z-disc to Z-disc. It's just anchorage for the thin filaments. You can see the I is here, and here we only have the thin filaments. The A is all of this, the overlap between the thick filaments and the thin filaments. Smack in the middle of the A band is the H zone, where we only find the thick filaments anchored to this M line here. You'll also see some elastin filaments that attach to the thick filaments and the thin filaments that help give us some of that elasticity that we see in our skeletal muscles and our cardiac muscles. So this is what a sarcomere looks like, and you guys are going to get to see a sarcomere three-dimensionally. We have a nice little 3D model, and then you guys are going to get to make your own models as well. So hopefully that will help you understand this structure. So if we go kind of from the smallest structure to the largest structure in terms of kind of how this all comes together, fibers within fibers within fibers. We start with the myofilaments. They get arranged in that sarcomere structure, and that makes up the myofibrils, thousands of sarcomeres. The myofibrils get grouped together, and that becomes a muscle fiber or muscle cell. That muscle cell, muscle fiber, gets surrounded by the endomysium membrane. We get a group of muscle fibers coming together. It's a fascicle. The fascicle gets covered in perimysium, and then we get a bunch of fascicles grouped together, and we get the whole muscle covered in epimysium. And so it really is this, from this very small structure, fibers within fibers within fibers. This is table 9.1. It's a nice little overview of these different membranes as well. The other thing that you'll see when you look at a skeletal muscle is you'll see a very, very, very well-developed sarcoplasmic reticulum. And what we mean by sarcoplasmic reticulum is it's just endoplasmic reticulum of a muscle cell. So it's smooth endoplasmic reticulum, and its primarily function here in muscle cells is storing and releasing calcium, because calcium is incredibly important to the contraction of our muscles. It's very, very, very well developed. It's really complicated and complex, covers the whole muscle cell, and it interacts with these transverse tubules, or T-tubules, which are basically indentations of the sarcolemma membrane that kind of really delve into the sarcomere structure and kind of pair with what we call these terminal cisterns. The word cistern just means well, to form a triad. And this is where the calcium kind of gets out of the sarcoplasmic reticulum and into the muscle cell, so it can actually do its job. So here you go. Here's your very well-developed sarcoplasmic reticulum. Here you can see these indentations, these two T-tubules, and these wells or cisterns on either side of the T-tubule makes up what we call a triad, because we have two cisterns, two wells, plus the T-tubule. And this really is functioning in storing and releasing calcium into the muscle cell so that the muscle can do its job. And in terms of how muscle contraction works, it's really the contraction of the sarcomere that is actually responsible for the overall contraction or shortening of your muscle. So we get these connections between the actin and the myosin when there's calcium involved, and ATP, of course. And the sarcomere then shortens such that basically more and more of the muscle fibers, more and more of the myosin and the actin overlap. So the actual length of the specific filament itself aren't changing, but what's happening is more of it is overlapping. More myosin heads are attaching to actin and kind of pulling it in more towards the middle. And you'll be able to see that really well with the big model that we have in class.